Welcome to the first episode of Two in a Billion, a podcast under the series of Joy of Science by Research Matters Gubi Labs. On this series, we ask questions about scientific research to scientists across India. I am your host, Aishwarya Viswamitra, and I am a biology major at Indian Institute of Science, Education and Research, IISER Mohali. Today we have joining us Dr. N.G.Prasad, an evolutionary biologist at IISER Mohali. His model organism is Drosophila, and today we're going to ask him questions about his model organism, the fruit fly, and about research he conducts using it. Hi, Dr. Prasad. Thank you for being on the show.
Hi, Aishwarya. Thank you for having me on the show.
Drosophila is the scientific name for a fruit fly.
Yes, it is.
And these fruit flies you see hovering over fruits at home as well.
Yes, absolutely.
So, as a child growing up, my first instinct would be to kill these fruit flies or to,
Yes.
or to get them away from fruits.
Yes.
Did you have to deal with some sort of disgust when you started working with insects? And did you have students who did?
Interestingly, I don't remember ever being disgusted with fruit flies when I started working with them. Probably because in the lab, the kind of conditions under which they are maintained are very different. They grow up in pre-made food, which is well calibrated. You cook it for them. Then they are grown in these nice glass vials or bottles. They're maintained in neat-looking incubators. When the larvae grow up, the pupation happens. When the adults come out, you handle them in a certain way. So at no point of time would you encounter anything that is disgusting. I have worked with the flies myself for a long time. I have worked with many students who have had to work with flies. By and large, I have not met anyone who said that, oh, this is disgusting.
Right.
That's it. People say that Drosophila labs smell. That is true.
I agree with that.
Yeah, they do smell. They do have the typical smell of the Drosophila food, which is, banana and jaggery and yeast and all that. They have a certain smell. That is something that I used to recognize and register maybe the first 2-3 weeks of my scientific career. After that, I don't even register it.
Nose blindness.
Nose blindness, yes.
Why did you choose Drosophila?
It is basically because of the field that I'm interested in. What I'm interested in is trying to understand how evolution happens on a real-time scale. If I have to observe evolution happening in a real time, then I need a system that I can deal with in a lab. Drosophila offers me all those opportunities.
Right.
It is genetically highly variable. I can easily culture it. And it has a very short lifespan. Right? It can go from one generation to the next in about 2 weeks' time, which means that I can have about 26 generations in a year. And for real-time evolutionary studies, you need to work with flies for a really long time in terms of number of generations. Therefore, Drosophila for me works very well. And therefore, Drosophila. It is, to me, a model which is very convenient to work with.
So, can you just give us an example of any study that has happened in your lab?
Sure. One of the primary studies that happen in our lab is about the co-evolution that happens between males and females in a sexually reproducing species. Most of the times when you talk about sexually reproducing species, the common idea is that males and females have to cooperate in order to reproduce, which is true. That has to happen. But to a great degree, there is also a lot of antagonism between males and females. And this antagonism has its roots in the basic difference in the reproductive investments that these two sexes do.
Exactly.
Typically, females invest a lot more in reproduction and typically males invest a lot less in reproduction.
And this is because females' eggs, they have to produce more.
Yes. And, in other organisms, there can be other things like, maternal care, the placenta, lots of other investments can go into it, or lactation, which can be very, very energy intensive. In general, theory suggests that females invest a lot and males invest very little, which means that the way the two sexes maximize their fitness is very different. And quite often not just different, they are completely opposite to each other. So over many, many issues, males and females are in conflict. In Drosophila, one characteristic trait is something called mating rate. How many mates do organisms have in a lifetime to maximize their fitness? For males, more the number of mates, it's better. For females, that's not the case. So over mating rate, there is conflict in males and females. Now this idea of conflict between males and females, what is called intersexual conflict, had been theorized for a very long time. People had also done some experiments. Our lab has done a detailed set of experiments using what we call laboratory experimental evolution approach, where we created populations in which either this male-female conflict was made to be very high or maintained at a very low level. This we achieved by manipulating operational sex ratios. We maintained certain populations at more male bias sex ratios when this conflict becomes very high, or female bias sex ratio when the conflict becomes very low. We let them evolve. As of today, they are about 250 generations old.
Wow.
And every few generations, we have looked at various characters in these populations. And I can show you a lot of characters that have evolved, and we have seen it evolve in real time. Wow. So we have been able to gather real-time evidence about intersexual conflict using Drosophila as a model system. That's one of the primary studies, one of the primary interests of the lab.
In tracing back to what you had said, this is only possible because of their short lifespans.
Absolutely, absolutely. 250 generations is still about 10 years, but
That's
you can still do it. Impressively, there are populations, not just in our lab, but around the world, which have undergone 1,000 generations of evolution.
Wow.
So these are sort of heritage populations that are maintained for really long term, for an entire career of a scientist, and probably passed on to the students to take care of. And they have yielded an absolute gold mine of information. It's a very powerful technique, very painstaking, but very, very insightful.
And this is probably why Drosophila research is done, because there are so many decades of research backing it up.
Absolutely, yes. We know so much about Drosophila. It could be the basic genetics of it, of late the more molecular biology of it. We even know its entire genome now. It has been sequenced. We know a lot about Drosophila development. And again, over the last many years, we have learned about its behavior, its evolution, and therefore it offers an extremely powerful system with which you could address extremely difficult questions. There are not many systems which will give you all these advantages. So therefore, Drosophila turns out to be very, very useful.
How do you get the fruit flies in the first place? I can't imagine you run out into the field catching them with nets.
Right. See, how do you get the fruit flies depends on the question that you're trying to ask. Suppose my question is, I want to study some character, let's say distributed along a temperature gradient. I want to find out what happens as to body size as temperature changes. So maybe one way to do that is to go to a temperature gradient. We are very close to the Himalayas, so maybe I will sample starting from the bottom of the Himalayas, go to higher regions, at each region catch flies, look at them and answer the question. In such studies, you do actually go out and do collect wild flies, bring them back and do studies.
Oh wow.
That's done.
Okay.
For certain other studies, let's say purely genetic studies, what one would call, the Mendelian kind of genetic studies, yes. You would not do that because you want to be able to say that I know everything about the genetics of this. So therefore, when I take 2 flies and make a cross, I know exactly what I am doing. So these flies have been kept in the lab for a very long time, and using a technique called inbreeding, you have made sure that all the flies in that population are identical, genetically speaking. So there you are not going out and collecting flies every day. For the kind of work I do, I need populations which are genetically variable but are adapted to the laboratory conditions. They are like my zeroth line. I control carefully all the environmental conditions, be it light, food, temperature, humidity, everything is controlled. And then this system, because everything is controlled, gives me the opportunity to change one or two environmental conditions of my choice. And then I can see the populations respond. And because I am controlling everything, when I see the response of the populations, I know that this response is because of the factor that I changed and nothing else.
Because you know everything about the fly already.
Exactly. So therefore, the flies that I work with in my lab are what are called long-term laboratory adapted populations. They were brought into the lab at some point of time in the wild, and ever since then, they have been maintained under constant conditions. Some of the other flies that we have in the lab were collected in the 1990s. There are some that were collected in the 2000s, but they have all been in the lab for more than 10 years, some 20 years, some 40 years, under identical conditions. So, for my kind of studies, I do not go out and collect the flies every time. There might be some other study where this is necessary to be done, but otherwise, no, not for my kind of studies.
Got it. So, minimize the surprises.
Yes.
So that you know that you have created the change.
Exactly.
So, talking about minimizing surprises, have you ever had flies escape?
Oh yes, they are very active. In fact, here is a hypothesis. See, when you bring the flies in and you are trying to culture them in the lab, there is a variation in the behavior of the flies. Some are very agile.
Of course.
Some are not so agile. And when I culture them over many generations, it's quite possible that some of them actually escaped. So the ones that I actually have in the lab, it's quite possible are the ones who are not so agile.
So you have the lazy ones.
You have the lazy ones because they failed to escape. So I would not be surprised if flies do escape. But if you ask me, most of the flies are well adapted to a laboratory condition right now.
Okay.
So they have been under 25-degree temperature, they get a certain kind of food, humidity, all that. They will not survive outside a laboratory environment for long.
Got it. From what you've explained, flies seem pretty easy to work with. Everything is established, temperature, food, you can store them in vials, and it seems pretty easy to work with. Do you think the common person could work with flies on their own, and do you approve of that?
I have two levels of answer for that. One is, flies are very convenient to work with. They are notoriously difficult to work with.
Okay.
They demand a lot of devotion and labor. It's like taking care of small children. They dictate your life.
Okay.
For the kinds of experiments that we do, for example, if the flies decide to come out of their puparium in the middle of the night, you better be awake, sit there in the middle of the night, welcome them to the world and start your work.
Wow.
If they decide to come out on a Saturday or a Sunday or a holiday, well, there goes your Saturday or a Sunday or a holiday, you be there and you work. So they do require a lot of work. So I don't think that, if I wanted to do something like what we are doing, I couldn't do it at my home.
Got it.
It's not possible. Yes, they are a nice organism with which you could teach, let's say, high school kids or primary school kids some basic biology. They're very nice. You can culture them very easily. You can culture them on things like banana or apple or very simple food, like the suji halwa that we make is essentially when you add a stabilizer, it becomes your Drosophila food. So you can actually do it. They are a good model. They are easy in that sense to maintain. However, I would say I would discourage people from just going out and catching them and bringing them back. See, whenever you use an organism in a lab, you need to have a certain basic respect towards that organism.
Absolutely.
Right? You're doing experimentation with a living organism. I wouldn't advise anybody to go and collect any insect and bring it back and mount it and or try and maintain it at home if you're not sure of what you're doing. Go out, Go to a Drosophila lab. There are so many around the country. Learn something about them. And after that, decide whether you want to do it. But generally going out and collecting insects, I wouldn't advise that.
In your lab, there are people from all subjects in IISER Mohali who
Yes.
end up in your lab for a procedure
Yes.
called for fly counting.
Fly counting, yes.
Tell us a little bit about that.
I think it sort of happened fairly organically when I started setting up my lab at IISER Mohali. Bodhisattva Nandi was a PhD student. He was very enthusiastic. He loved interacting with undergraduates. And undergraduates are our greatest strength in an IISER system. We could not have done most of the things that we have managed to do as a lab if there were no undergraduates here. IISER undergraduates are brilliant, very bright, very motivated. They don't have to do most of the research that they do, but still they come to the lab, they do the research because they are interested. It's not part of their curriculum, but they do it out of the interest, and that's the best thing that you can have. So when we started the lab, what happened was we had similar undergraduates who were very, very interested. There was Abhilasha, there was Anshu, there was Keshav, Vikesh, Manish. Each of them had a different majors in mind.
Okay.
Abhilasha and Mehreen took biology, Anshu, Vikesh took physics, Manish took chemistry, Keshav was a mathematics person. But they would all sit in the lab, count flies, do evolution experiments, and then go back and study whatever their subjects were. I think it all boils down to what kind of an environment you have in the lab. And since these are very bright kids, as long as they are getting something new to learn and the environment is fun enough for them to spend time in, they will come, they will work because they like it. And on a very practical note, a lot of these people who might never end up doing their further studies or research in biology do end up having papers in evolutionary biology. Pradeep, who is now doing a PhD in chemistry, his first publication is in the topmost journal of my field called Evolution, because as an undergrad, he did a lot of experiments which were so good that they finally ended up in evolution. So you do have all these very interesting crossovers. It does happen.
That's lovely. And that again just speaks about how science is becoming very interdisciplinary now.
Yes.
And sharing is of the utmost importance.
Absolutely, yes.
So thank you for sharing all of this with me, Dr. Prasad.
It's been great, Aishwarya. Thank you so much.
Thank you.
And thank you, listeners and readers of Research Matters. For more such podcasts and science news, log on to researchmatters.in.
And while you may still want to swat the next fly you see, don't forget that life around you is beautiful and that it was created by nature just like you were. Keep asking questions and keep spreading the joy of science. I'll see you on our next episode.